Welcome back to the first lesson of the biotechnology and unit. We're talking about bacteria. We're going to start off. We just got done talking about transformation, where some other genetic material can be put into another cell, and then that cell can use it as its own. That's transformation. And the Griffith experiment, which I kind of explained pretty quickly before, because we did it before, shows that. So let's go to transduction, which is the second way. This is the process where viruses can carry prokaryotic genes from one bacterium to another. This picture over here shows how Hershey and Chase showed that DNA was actually the genetic material, not proteins, but DNA. So let's look at transduction. So this is a virus, and what happens with a virus, it's called a phage when you're dealing with bacteria. It'll carry its DNA and put it into a bacteria. Then the bacterial processes will make new copies of the DNA. Okay, the bacterial DNA gets broken apart and it begins to make new virus DNA. Well, after that host cell makes new virus DNA, it's going to make new viruses. As you can see here, new viruses are made. Those viruses can then break off and go infect other cells. But sometimes the viral DNA doesn't get put into the virus, but the bacteria, see how the bacteria's DNA, the A positive part of it, gets put into this virus instead of the actual viral DNA, or RNA. It's usually RNA, not DNA. But and then that virus can go travel to another cell and put its, well, not its, but that old host cell's DNA into a new cell, thus giving that cell some new DNA, that bacteria some new DNA. And this is a picture showing how the whole thing would happen. All right. Understand that with this transformation and this transduction, we're trying to show you how bacteria can become very diverse very quickly. You get all these new ways to put new DNA in them, and that DNA can be used to help fight off antibacterial methods. The third method, conjugation. Some prokaryotes can create what's called a mating bridge. And that mating bridge can transfer DNA from one bacteria to another. It's a kind of a sexual reproductive way. Not technically sexual reproduction because you're not reproducing a new organism, but it's kind of like a way to sexually recombine genes. And that's conjugation. Excuse me. Look here, I jumped ahead. And this picture, this slide just kind of shows that. Here is the mating bridge. Here is a plasmid. Plasmid's another word for bacterial DNA. And it can be put into the cell, and that cell can then have that kind of DNA. And there's two different ways. You could have its own F plasmid, or you can have kind of a put-in recombinant type of section of gene that can then make the mating bridge and have it go across. Remember, students, don't forget when you're looking at it to read the notes at the bottom. So there's three ways of horizontal gene transfer. We've gone over transformation, which is the DNA coming into the cell. There's transduction, where a virus puts it in, and there's conjugation, where it's done over a mating bridge. The final way, transposition, is when genes jump. So you can take this gene right here in the DNA, you can make a copy of it and have that gene implant in a new part of the DNA, which can actually change the genetic makeup of the bacterium and maybe make it do some new things. And that's what's happening here. It's kind of like a copy and paste. And in 1983, at the age of 81, Barbara McClintock won the Nobel Prize for discovering jumping genes. But she didn't, back when she was doing the work, women were underrepresented in science and sometimes didn't get the credit they deserved. So at least she was able to get this during her lifetime. And that concludes the first lesson. Good luck, and see you soon.